Our next speaker is Alessandro Baldi-Tallini from PerkinElmer. During his 30-plus year career, Alessandro has always been involved in chromatography and mass spectrometry, first as a researcher and customer, and later as an active member of the analytical instruments industry. He spent 12 years at PerkinElmer, where he covered several roles in the chromatography and mass spectrometry business units. Later, he joined Protea Biosciences as general manager. At Protea, Alessandro drove the go-to-market strategy and execution of an innovative device for imaging mass spectrometry. After 3 years at Protea, he became VP of Strategy and Business Development for the holding that controls Dani Instruments, and eventually became CEO of Dani Instruments USA. Recently, Alessandro joined PerkinElmer again as business line leader of mass spectrometry. And so, without further ado, I'll turn the presentation over to Alessandro Baldi-Tallini. Alessandro?
Thank you, David. Good morning, good afternoon, everyone. Today, I'd like to cover the analysis of contaminants in food matrices performed by triple quad HPLC mass spec. And in particular, I would like to talk about some specific food safety matrices like wine, milk, coffee, or potato chip. So we will talk about the principle of HPLC mass spec and the role in the application in food safety, and I will cover some of the latest development of PerkinElmer flow-based triple quad mass spectrometry in matrix like wine, coffee, milk, or potato chips. And then in the end, I'd like to talk about some of the features of flow-based mass spectrometry and its advantages. Food safety is a science, and the application of triple quad mass spectrometry has become more and more important, giving important contributions to this science. It's an entire value chain, it's an entire workflow that starts from extraction and cleanup down to the separation in HPLC, and then the detection and the mass spec analysis, identification and quantification of compound, and the final reporting. The most relevant analysis for HPLC mass spec are in the domain of antibiotic testing, mycotoxin and toxin in general testing, as well as pesticide analysis. Why HPLC mass spec has become so relevant, so important? We all know that in a food safety analysis, HPLC mass spec is used as a quantitative tool to measure the amount of a certain molecule in a certain food matrix. And we all know that we operate in LC mass spec in multiple reaction monitoring, or MRM, using the first quadrupole as a filter and the third quadrupole as a filter, and using the second quadrupole or hexapole as a collision cell to generate a fragmentation. But why we are able to reach such a high level of sensitivity with LC mass spec? The reason is that during this analysis, we are actually applying three different processes. We are separating molecules based on time, and this is based on chemical interaction. We are using mass separation, that is a chemical formula based, and we are using fragment separation, that is based on chemical structure. We all know that when we apply orthogonality to a chemical analysis, we can reach a very high level of specificity. And if we reach high level of specificity, of course, we will be able to reach a very high level of sensitivity. So it's the combination of three different principles: time separation, mass separation, and fragment separations, that are giving HPLC mass spec the power to become a very important tool for quantitative analysis. So we are introducing the PerkinElmer QSight triple quad mass spectrometer. And this device is based on what we call the flow-based mass spectrometry. That is a way to manage ion in the upper part of the mass spec workflow. So we are using a coaxial flow electrospray to improve desolvation and to improve sampling of charged species. We are using an innovative Stay Clean source that is an auto-cleaning source to improve sensitivity and to enhance instrument uptime, to provide exceptional instrument uptime. And we are also moving ions with a laminar flow into the ion guide from the source down to the interface, down to the mass spec, using just a flow-based mechanism. So the flow-based mass spectrometry is providing us sensitivity, is providing a high instrument uptime that, combined with the fast acquisition speed of the mass spectrometer itself, allows us to run many compounds per run, to analyze many compounds per run, and to run many samples per shift, and in the end to provide a very high level of laboratory efficiency. So I'd like to move on to some of the applications that we developed using this flow-based mass spectrometry system. The first application is the direct analysis of glyphosate in wine. As you know, glyphosate is a very controversial herbicide, and it's defined by the World Health Organization as a probably carcinogenic molecule to human. And recently, in April 2018, the European Food Safety Authority released a reasoned opinion reviewing the existing maximum residue level for glyphosate in different matrices. And glyphosate is very ubiquitous. It can be used, for example, to clean walking area, and it's used for many different crops, and so as such can be found in many different food like beer, wine, honey, or infant formula or breakfast cereals. So it is a very polar compound, relatively difficult to analyze, but is very important, as can be really found in many different matrices. So we developed a very effective method to allow direct analysis of glyphosate and its metabolite AMPA using a very simple sample preparation. And so this is also a way to show the capability of the PerkinElmer QSight system to work with dirty sample matrices. What is the fastest way of preparing a sample if just no preparing the sample? So we are just taking a wine in this sample preparation workflow. We are filtering the wine with a nylon filter, and then we spiked this sample with standard, and we did the injection and the analysis in the UHPLC mass spec system. So we were able to build a calibration curve for AMPA and glyphosate, and the calibration curve was ranging from 6 parts per billion up to 200 parts per billion. And again, this is just the analysis of glyphosate and AMPA in red and white wine with just a filtration of the sample using a nylon filter. So the sample is relatively dirty, is wine as is, so we wanted to check the robustness of this method, performing almost 200 analyses of glyphosate in red and in white wine. These are the result of the variability of this 200 analysis of a wine spiked with 50 parts per billion of glyphosate and directly injected into the UHPLC mass spec system. And as you see here, the variability is around 10% and 8%, so it's a very low level of variability that demonstrate how the system can really handle dirty sample over time, sample after sample, day after day. The next example is on antibiotics analysis. Antibiotics are also used very often for treatment of animals or prevention of animal diseases or as a growth promoters. So it can be found in meat, for example, or in milk, but also in the product, in the byproduct of milk, for example. And so the tolerance level of this product in milk are relatively high. Let's say that they are relatively easy to analyze by LC-mass spec. And so if you can see here on the right, we are talking about the 5, the 10 parts per billion for each different antibiotics, with some exception of tetracycline, chlortetracycline, that have a much higher tolerance level. So, as I said, it's relatively simple to match this level, but we wanted to go down and explore how low we can go, and the reason is that milk is a matrix that is often used after concentration. So let's think about milk powder, for example, or it's used for dairy production. And as you know, dairy is a process that is using enzyme or is using bacteria, for example, for cheese production. And of course, these bacteria cannot absolutely tolerate the presence of any antibiotics because the process will be completely inhibited or will go into a different direction. So other bacteria may be resistant to these antibiotics, will take the lead and will produce a product with characteristics different than expected. So before doing the analysis of UHPLC mass spec, we perform a sample preparation of the milk, an extraction of the milk. The extraction of the milk is a modified version of the QuECHERS method, where we added a protein precipitation step, but then after some centrifugation, we did some sample cleanup, defatting of the milk with the dispersive solid phase extraction. And then after some centrifugation and reconstitution of the sample, we did a last filtration and a simple injection of the extract of the milk into the HPLC mass spec. For each antibiotic, we defined a quantifier ion and 2 qualifier ions to confirm the presence of that specific molecule. So this is an example for cloxacillin and the MRM experiment at 5 parts per billion in milk extract. And in this next slide, there is another example of sulfadimetoxin MRM chromatogram at 5 parts per billion in milk extract. These are the results of the analysis, where we can see that we found a very low limit of detection and limit of quantitation. So we were able to go sometimes 10 times up to 300 times lower than the tolerance limits for each antibiotics. And again, this is the level of sensitivity that the cheese producers, for example, want to see when we do a milk analysis for antibiotics. Since the milk extract is a relatively dirty matrix, we also perform a stability testing, a robustness testing, using some of the antibiotics, and we analyze the same milk extract for 6 consecutive days. As you see, the standard deviation of the results is extremely good, so the instrument is showing a very high level of robustness and resistance to contamination. This is ciprofloxacin, and a similar result is for sulfametazine at 10 parts per billion. This is again an analysis for 6 consecutive days, and the standard deviation for this molecule for almost 500 injections is 4.5%. So again, very stable result, very good performance from an instrumentation standpoint. So just to recap, we were able to analyze many different antibiotics from 5 different panels of milk, and we were capable to reach a level of sensitivity that are from 10 to 300 times lower than the tolerance limits of each antibiotics. The next application we would like to talk about is related to acrylamide. As you know, acrylamide is formed during the Maillard reaction when a starchy food is exposed to temperatures that are higher than 120 degrees centigrade, and as a byproduct we have the formation of acrylamide. Recently, the European Commission regulation confirmed that the previous evaluation of acrylamide in food can potentially increase the risk of developing cancer for consumers in all age groups. So the current level of dietary exposure is actually a concern with respect to its carcinogenic effect. So for this reason, the European Community, as of April 11, 2018, forced the food operators and all the people that are doing food analysis to analyze for presence and level of acrylamide in the foodstuff that is part of a list of the Annex 3 of this regulation. This, of course, generated a lot of attention and a lot of visibility to the acrylamide problem, and in particular, since coffee can also contain acrylamide because the roasting of the coffee is indeed a Maillard reaction, and coffee is indeed a starchy food to start with. A new attention was posed on the content of acrylamide in coffee, and for instance, the state of California released a regulation where in the near future there will be a warning for consumer about the presence of possibly cancer-causing chemical in the coffee. So the European Community defines some benchmark references expressed in parts per billion or microgram per kilo, and in particular for roasted coffee it is 400 parts per billion, and for instant soluble coffee it's 850 parts per billion. So what we did an analysis of coffee beans, and we developed a sample preparation taking half a gram of coffee, of grinded coffee powder, and then doing a methanolic extraction, and then a filtration in a centrifuge, evaporating the solvent, and then putting the sample in water, and then doing a defatting analysis with a solid phase extraction cartridges, and then we analyze the eluant from this cartridge.